presentation, also from a national center of research from Switzerland, and the speaker is Barbara Rothen Rutishauser, and she comes from the University of Fribourg. Okay, I hope it works. So the floor is yours.
Thanks for organizing this very interesting session, and thank you for coming so numerously to this very good hidden lecture room. So I hope that I can maybe give some more answers to the questions which have just been asked, because I will focus in my presentation on quantification, on intracellular nanoparticles in single cells. So when we design nanoparticles, which might be used ones for biomedical applications, we usually do very simple cell culture experiments to assess their toxicity, immune reactions, and uptake. And for this, of course, we dilute the particles in cell culture media. This is a completely other topic, which would take a lot of time to talk about characterization of nanoparticles in this cell culture media, and then also actually how much particles deposit on the surface of our cells. And this is then what I will talk about today, is how can we assess how many particles are attaching to the cell surface, how they are taken up, as we have just heard in the recent presentation, and of course also then how many particles are intracellular. I will not talk about uptake mechanism, but just give a very general presentation about intracellular nanoparticles. These are some examples of nanoparticles which are used mainly in nanomedical applications. Of course, we have a lot of spherical particles like gold nanoparticles, silver nanoparticles, iron oxide nanoparticles. But then we also have very different shapes. These are gold nanorods. We have hybrids, so particles which are made of different materials. And then we also have liposomal delivery systems, which is a completely different material. And then as soon as you go into biology or medicine, then people ask for fluorophores, because we have a lot of quantification methods based on fluorophore detection possibilities. So we can attach fluorophores to the surface of gold nanoparticles. I will talk about this also in more detail later. Or we can integrate a fluorophore in a silica shell and make the particles then fluorescent to detect them by fluorescent microscopy. So these very different particles already point to the issue that we need also very different methods to quantify and to analyze them. You don't need to read it. I just have tried to summarize some of the most often used techniques, which are based on chemical analysis, which can be used, of course, for metal oxides, magnetic nanoparticles. Here we have a very low sensitivity, but we do not have spatial information, so we do not know if particles are attached to the cell surface or if they are intracellular, which is important for their possible induction of cell responses. Then we have fluorescent-based quantification techniques, which depend on a fluorophore, but we could also use quantum dots. Here, of course, we have spatial information. We can use high-throughput methods. But if we work with fluorophores, we need to be aware that it's only semi-quantitative, because fluorophores can, of course, bleach. They can also detach from the particles. And we have a very low resolution, although we heard this morning in the keynote lecture, if you use specific techniques, we can increase this resolution. Then we have electron microscopy methods, which needs electron-dense nanoparticles. Of course, the resolution here is very high, it's very good, but if need, or if we would like to do quantification, we need some very complex technical procedures, sample section preparations, and stereological approach for quantification. There are a lot of reviews out. I just found a very interesting review from Castillo and his collaborators, which has been published some months ago, which really gives a very nice overview about recent techniques. We also have a lot of new techniques, of course, available, which I do not have the time now to go into it. Let me give you one specific example and experience we did in our research lab when we tried to investigate uptake and also quantify nanoparticles. So in the NRP64 project, which has now already been stopped since some months, we have decided to design fluorescence-encoded gold nanoparticles in order to follow up their path inside the cells, but also when we use them in vivo, and if we can track different immune cells with these particles. As I just said, for biological methods, we need to attach a fluorophore to the gold core and also a polymer to make them colloidal stable. So we have used polyvinyl alcohol, which is just one possible polymer. And then we have induced or attached the fluorophore inside the polymer by also attaching a second layer of polymer in order to shield the fluorophore, the properties, and to protect the fluorophore for possible degradation. We have characterized the particles in complete cell culture media to be sure that the particles are stable over 24 hours when we add them to the cell culture media. We also did some microscopic pictures, and we could really show that single particles attach to the outer cell surface during these 24 hours. And to do some first experiments, we have used the macrophage cell line just to investigate particle uptake in one possible immune cell over time. So when we then started to do fluorescent to use fluorescent quantification techniques like flow cytometry, or here I show laser scanning microscopy, and we try to compare different charged particles negatively versus positively charged particles, both labeled with the same fluorophore. We could see indeed some uptake for the positively charged particles, but no uptake for the negatively charged particles. And then also in our in vivo experiments, we could only see the positively charged particles, and then people, of course, to the conclusion negatively charged particles do not enter cells. However, I do not believe in this hypothesis, because I personally think that each particle which is attaching to the cell surface is somehow moving on the cell surface and then taken up. So I've asked my PhD student to look into other techniques, and we have used dark field imaging microscopy, and indeed here we see negatively charged particles inside the cells. In addition, we also did electron microscopy, and here you see some vesicles, maybe endosomes, and here you see these gold nanoparticles inside the cells, negatively charged and also positively charged particles. We also did then ICP, so inductive coupled plasmonic spectroscopy, where we could quantify the gold which is attached or intracellular, and we also followed this uptake over time. Here in red you see the positively charged particles, and what is really astonishing, after 24 hours we even have more negatively charged particles inside the cells. However, here we cannot really determine which particles are still outside and which are inside, so we need to do more electron microscopy. So why can we not see the fluorophore here for the negatively charged particles? We did then some experiments with artificial lysosomal fluid, which is a solution which you can prepare. It's a low pH, containing a specific composition of ions, and enzymes. And when we did cryo-electron microscopy, we could see that for the carboxylated particles, we see some aggregation in this low pH environment, which was not the case for positively charged particles. So this is also obvious if we look macroscopic, so we can see that these particles tend to aggregate, and this aggregation inside the lysosomal fluid give us some indications that the fluorophore, the polymer, seems to be destabilized. I do not say it's degraded, but destabilized, and this could induce a quenching of the fluorophore. So these particles are taken up, but we cannot see them by fluorescent methods. So to conclude, we could show that also negatively charged particles are taken up to a very high concentration if we compare to the same particles with a positive charge, and we have some indications that they aggregate inside the lysosomes, and this can induce a quenching of the fluorescence. So to summarize, quantification is really a challenge because of these very different materials, these very different properties, high reactive surface that these particles have. And it's really important, as already has been said in the previous presentation, we need to combine different methods and also to include controls, a lot of controls, and I usually ask the students to do more and more controls, but it's important that we do not interpret any artifacts. With this, I would like to finish and thank my group, all the collaboration partners, the foundations for their support, and you for your attention. Thank you.
Thank you very much. You are also very well in time, so I really appreciate that you have shown that one has to verify the results. I think there are some questions.
Hello. So regarding the lipid nanoparticles, how you can quantify? These are the gold nanoparticles you can quantify.
I mean, one possibility is to attach a radioactive label, but lipids we cannot quantify inside cells. We would need somehow to integrate a metal particle, but lipids themselves we cannot quantify. There are, you can attach a fluorophore, then you can also assess the fluorescence, but the lipids themselves we cannot. Or do you know? No.
So for liposomes, you have to attach fluorophores, right?
So, yeah, or any other label, yeah.
Okay. Thank you.
There's another question in the back.
I mean, if you have doxorubicin, encapsulated doxorubicin, it's fluorescent, so you could indirectly track them.
Hello. So you showed that you attach a fluorophore to a gold nanoparticle in order to quantify its localization. But is there any chance that you can alter the properties of your nanoparticle by attaching the fluorophore? So how can you control that you don't change something?
We have done, of course, a lot of particle characterization methods, and when we have the fluorophore on the outer side, we inhibited the cell uptake. We don't know why. This is why we have added a second polymer layer to shield the fluorophore properties. But also fluorophores have a charge, a zeta potential, which can induce different cell uptake. Yes, very important.
Okay, I think there's no further question. Thank you very much. Thank you, the speaker, again.